Every other organ in your body has a drainage system, a network of vessels that runs alongside the blood supply, collecting the waste that cells produce, carrying it away, dumping it into the lymph nodes for filtering. Your legs have it, your gut has it, your skin has it. For more than a century, anatomists opened the human body, traced these channels through every tissue they could find, and arrived at a single strange exception. The brain had none. The most metabolically expensive organ you own, burning 20% of your energy while making up 2% of your weight, producing more molecular garbage per gram than anything else inside you, appeared to have no plumbing at all, and nobody could explain how it stayed clean. For decades, the textbook answer was that the brain simply did not produce much waste, or that whatever it produced diffused away slowly into the bloodstream. Both of those answers were wrong. The truth is that your brain is cleaned every single night by a system that did not have a name until 2012, that moves a volume of fluid through your skull that would have seemed impossible, and that switches on almost entirely while you are unconscious. You have been running this machine your entire life. You have never once felt it. And if it fails, the consequences are written into one of the most feared diseases on Earth. Start with the problem the brain has to solve. Every thought you have is electrical and chemical. Neurons fire, they release neurotransmitters, they burn glucose and oxygen, and like any engine that burns fuel, they leave residue behind. Among that residue are proteins, and two of them matter enormously. One is called amyloid-beta, the other is called tau. In small amounts, produced and cleared in balance, they are harmless byproducts of a working brain. But amyloid-beta has a dangerous property. The molecules are sticky. Given time and concentration, they clump together into plaques. And those plaques are the physical signature found in the brains of people with Alzheimer's disease. So the brain faces a continuous engineering problem. It generates a toxic, self-aggregating protein as a normal part of thinking, and it must remove that protein faster than it accumulates. The question that haunted neuroscience was simple. Without lymphatic vessels, how? The answer began to emerge in the laboratory of Maiken Nedergaard at the University of Rochester. Her team was studying the spaces around blood vessels in the brain, and they did something that older anatomists could not. They injected fluorescent tracers into the cerebrospinal fluid of living mice and watched, in real time, through the intact skull, where that fluid went. What they saw was not slow diffusion. It was a current. Cerebrospinal fluid, the clear liquid that surrounds your brain and spinal cord, was being driven down into the deep tissue of the brain itself, flowing along the outside of arteries, pushing through the spaces between brain cells, and exiting along the outside of veins, carrying dissolved waste with it. The whole loop was a circulation system hidden in plain sight, running through channels nobody had recognized as channels. But the part that made the discovery extraordinary was the engine that drives the flow. And here the brain does something no other organ does. It uses its own support cells as plumbing. Your brain is not made only of neurons. More than half of it is glial cells, and the most abundant are astrocytes, star-shaped cells that were long dismissed as mere scaffolding. Nedergaard found that astrocytes wrap their endfeet around every blood vessel in the brain, forming a continuous sleeve, and studded into those endfeet are water channels called aquaporin 4. These channels act like one-way valves that let cerebrospinal fluid pour out of the space around the arteries and into the brain tissue, sweeping waste toward the veins. Because the system depends on glial cells and mimics the function of the lymphatic system, Nedergaard named it the glymphatic system. Glia plus lymphatic, a drainage network that runs not through dedicated vessels, but through the living architecture of the brain's own helper cells. When her team deleted the aquaporin 4 channels in mice, clearance of amyloid-beta dropped by about 70%. The valves were not decoration. They were the entire mechanism. Remove them and the garbage stays. The reception was not immediate acceptance. When Nedergaard first proposed that a brain-wide fluid circulation existed and that astrocyte water channels powered it, parts of the field were skeptical, and some still argue about how much of the movement is true directional flow versus ordinary diffusion. This is not a footnote of academic politics. It is how a genuinely new idea enters science. The brain had been studied for 2 centuries with the assumption that it had no waste clearance plumbing, and overturning a 200-year-old assumption requires evidence strong enough to survive sustained doubt. The aquaporin 4 result was one of those pieces of evidence, because it did not merely describe the flow. It broke it. When the channels were gone, the clearance collapsed. And a mechanism you can switch off and watch fail is a mechanism that is real. And amyloid-beta is not the only thing the tide carries. The other protein, tau, is in some ways more dangerous, because where amyloid clumps into plaques between cells, tau tangles up inside the neurons themselves and appears to spread from cell to cell along the very circuits the brain uses to think. In 2019, a team led by Jerry Holth and David Holtzman at Washington University in St. Louis showed that a single night of sleep deprivation raised tau levels in the cerebrospinal fluid of healthy people, and that disrupting sleep accelerated the spread of tau in mice. 2 proteins, 2 diseases of aging, both cleared more efficiently in deep sleep, both rising when that sleep is cut short. The pattern is too consistent to be coincidence. Now here is where the story turns from anatomy into something that touches your life every night. Because the glymphatic system is not always running. It has an on switch, and the switch is sleep. In 2013, a researcher named Lulu Xie, working in Nedergaard's lab, published a paper in the journal Science with a title that sounds almost too clean to be real: Sleep Drives Metabolic Clearance from the Adult Brain. Her team measured the actual physical space between brain cells in mice that were awake versus mice that were asleep, and they found something that should not have been possible to miss for so long. During sleep, the space between neurons expands by roughly 60%. The brain cells physically shrink, the gaps between them widen, and cerebrospinal fluid floods into the enlarged channels. The result was that clearance of amyloid-beta during sleep ran roughly twice as fast as during waking. 2 times the cleaning, simply from being unconscious. Think about what that means. The brain cannot fully clean itself and think at the same time. The same channels cannot be open for high-resolution computation and wide-open drainage simultaneously. So evolution made a choice. During the day, the channels narrow, the fluid slows, and the brain prioritizes processing the world. At night, the cells contract, the channels open, and the brain switches into maintenance mode. Sleep is not the brain shutting off. Sleep is the brain changing jobs. And the molecular trigger for that switch is a single neurotransmitter you have heard of in a completely different context: norepinephrine. During waking, norepinephrine is constantly released, keeping you alert, keeping astrocyte volume high, keeping the channels tight. As you fall into deep sleep, norepinephrine levels collapse. When Nedergaard's team artificially suppressed norepinephrine in awake mice, the brain space expanded and clearance sped up as if the animals were sleeping. The chemistry that makes you alert is the same chemistry that keeps the drains closed. To clean the brain must first let go of vigilance. There is a kind of biological honesty in that. You cannot stay on guard and be cleaned at the same time. But there is a second mechanism operating at the same time, and it was discovered not in mice but in living, sleeping human beings. In 2019, Laura Lewis and her colleagues at Boston University put people inside an MRI scanner and did something unusual. They recorded brain electrical activity and cerebrospinal fluid flow simultaneously while the subjects fell asleep. What they captured, published in Science, was a rhythm. During the deepest stage of non-REM sleep, large, slow waves of electrical activity sweep across the brain roughly once every 20 seconds, and locked precisely to those waves, in perfect alternation, came pulses of cerebrospinal fluid washing up into the brain. First, the neurons would go quiet together. Blood would retreat from the active tissue, and into the space left behind, a wave of fluid would surge. Electrical wave, then fluid wave, then electrical wave again all night long, a tide synchronized to the rhythm of unconsciousness. And the mechanics of that pump are worth pausing on, because the brain does not contain a heart of its own to push the fluid. So what drives the tide? The answer is that the brain borrows the pulse it already has. Every time your heart beats, the arteries inside your skull expand by a tiny fraction of a millimeter, and that expansion squeezes the fluid-filled space that wraps around them, pushing cerebrospinal fluid forward in little pulses. During the day, this arterial drumbeat does some of the work. But during deep sleep, a second and larger rhythm takes over. As each slow wave of neural silence sweeps the cortex, the blood volume in the brain drops in unison, and that retreat of blood pulls a fresh wave of cerebrospinal fluid in behind it. The neurons going quiet together is not just an electrical event, it is a hydraulic one. Their synchronized silence becomes the suction stroke of a pump. And the deeper and more synchronized the sleep, the stronger the pull. The brain turned the rhythm of unconsciousness itself into a piston. This is the part I find genuinely strange. The slow waves of deep sleep, the very signal that defines the most restorative stage of rest, are coupled to the physical pumping of cleaning fluid through the brain. The depth of your sleep is not just a measure of how rested you feel. It is a measure of how thoroughly your brain is being flushed. And as people age, those slow waves shrink. The deep sleep that powers the tide gets harder to reach, which raises a possibility that connects the whole story together. Because here is what happens to amyloid-beta when sleep is cut short. In 2018, a team at the National Institutes of Health, led by Ehsan Shokri Kojori, took healthy adults and deprived them of a single night of sleep, then used PET imaging to measure amyloid in their brains. After just one night without sleep, amyloid-beta in the hippocampus and thalamus rose measurably compared to a rested night. One night. Not chronic insomnia, not years of deprivation. A single missed night was enough to leave detectable extra plaque-forming proteins sitting in the brain because the cleaning crew never came on shift. The implication is sobering, and it runs in a loop. Poor sleep allows amyloid to build up. And amyloid buildup, accumulating in the regions that generate deep sleep, makes deep sleep harder to achieve. The disease may dig its own channel. The worse you sleep, the more you accumulate. And the more you accumulate, the worse you sleep. There is even evidence that how you lie down matters. In 2015, Lulu Xie, working again in Nedergaard's group, used dynamic imaging to compare glymphatic clearance in mice sleeping on their side, on their back, and on their stomach, and published the results in the Journal of Neuroscience. The lateral position, lying on the side, produced the most efficient waste clearance of the three. The finding is preliminary and has not been confirmed in humans, and it would be a mistake to turn it into a rule about how you must sleep. But it hints at something quietly remarkable. The most common sleeping posture adopted by humans and most animals may be the position that best lets the brain drain, as if the body, long before any scientist understood why, had already arranged itself for the night cleaning. Alcohol, by contrast, points the other way. High doses suppress the deep, slow-wave sleep that powers the tide, which is part of why a heavy night of drinking leaves the brain feeling foggy the next day, even after the alcohol itself has cleared. The fog is not only the poison, it is the cleaning that never happened. This connection is why researchers now talk about sleep not as a symptom of Alzheimer's, but as a possible contributing cause. For most of medical history, the disrupted sleep seen in dementia patients was treated as a consequence of the disease eating away at the brain. The glymphatic discovery flips the arrow. If clearance happens mainly during deep sleep, and deep sleep declines with age, then decades of slightly insufficient cleaning could allow amyloid to creep upward long before any symptom appears. By the time memory fails, the plaques have been forming for 20 years, and every one of those years contained roughly 365 nights during which the brain either did or did not run its full cleaning cycle. Now, I want to be careful here because this is exactly the kind of finding that gets oversold. The glymphatic system is real, and the aquaporin 4 mechanism is well established in animals. The sleep-driven expansion of brain space has been replicated. The coupling of cerebrospinal fluid waves to slow-wave sleep in humans is documented. But the leap from there to the claim that fixing your sleep will prevent Alzheimer's is not yet proven. The human glymphatic system is harder to measure than the mouse version, and some researchers have questioned how much of the bulk flow seen in animals translates directly to people. What we have is a powerful, coherent, mechanistically plausible chain of evidence, with some links forged in steel and others still being tested. That is what honest science looks like at the edge of discovery. The mechanism is established. The full clinical consequence is still being mapped. But even the established part should change how you think about a night of sleep. Consider the actual numbers. Your brain contains roughly 125 to 150 milliliters of cerebrospinal fluid at any moment. But you produce around 500 milliliters every day, cycling the entire volume several times over. During deep sleep, the glymphatic tide drives a substantial fraction of that fluid through the brain tissue itself, rather than just around it. The system moves measurable volume, clears measurable protein, and does almost all of it in the hours when you are least aware of being alive. The most important housekeeping your body performs on its most important organ happens in a state you cannot consciously enter, control, or observe. And there is a deeper elegance hidden in the design. The brain solved its plumbing problem without building new vessels at all. It repurposed the cells that were already there. The astrocytes that feed neurons, regulate blood flow, and hold the architecture together also moonlight as the night cleaning crew, opening their water channels when the norepinephrine fades. The cerebrospinal fluid that cushions your brain against impact also doubles as its rinse water. Nothing was added. Everything was given a second job. The system is invisible precisely because it is not a separate organ. It is the brain using itself to clean itself, on a schedule written into the rhythm of sleep. There is a human story buried in this discovery that is worth telling, because it shows how easily this was missed. For over 100 years, the smartest anatomists in the world dissected brains and found no lymphatic vessels, and they concluded the brain did not need them. They were looking for the wrong thing. They were looking for pipes. The brain had not built pipes. It had built a process, a flow through living tissue that only appears when the tissue is alive and asleep, which is exactly the condition under which no anatomist ever examined a brain. You cannot see the glymphatic system in a dead, dissected, drained specimen, because the moment life and sleep end, the tide stops and the channels collapse. The discovery required watching a living brain in real time, something the tools of the 20th century could not do. The system was hiding not in a place but in a state. It only exists while you are sleeping. So think back to the beginning, to the strange fact that the brain seemed to be the one organ with no drainage. It was never an exception. It simply kept its plumbing in the fourth dimension, switched on only at night, run by cells everyone had dismissed as filler, driven by the disappearance of the very chemical that keeps you awake. Every organ has a drainage system. The brain's just happens to be made of time and sleep. Tonight, somewhere in the first deep stage of your sleep, the norepinephrine in your brain will fall away. Your neurons will shrink by a fraction. The space between them will widen by more than half. A slow electrical wave will roll across your cortex. The blood will pull back, and a pulse of clear fluid will surge into the opened channels, carrying with it the sticky residue of every thought you had today. It will happen again about 20 seconds later, and again. Hundreds of times before morning, a tide you will never feel, washing clean the organ that is, in the end, the only thing you have ever used to know that any of this is happening at all. You do not have to do anything. You only have to let go of the day. The cleaning starts the moment you stop holding on.